Morning everyone. Today topic is plasmids and its types. First, what is plasmid? Is a extra chromosomal and self and double-stranded circular DNA molecules. And these plasmids generally found in mostly in here the prokaryotic organism is bacteria. But there is one exception in eukaryotes also present. The eukaryotic organism here fungi. In fungi, the best example organism only yeast. In yeast only we have seen these plasmids, but mostly majority the plasmids are present in prokaryotes, that is bacteria. The key feature of plasmids, they replicate independently of the genomic or chromosomal DNA. So they do not depend any other chromosome. They replicate itself, means independently. And here the significance of these plasmids. Significance. While they are not essential for the basic survival. Generally, these plasmids are not essential for basic survival under normal condition. They are not essential for basic survival under normal condition. But they provide unique phenotypic character, very important. But they provide unique phenotypic character to the host bacteria. So that is best example. These plasmids carry anti-resistant characters. They carry. So this is the significance of plasmids. Use of plasmids. Generally, plasmids are used as a cloning vector in rDNA technology. Used as cloning vector in rDNA technology, recombinant DNA technology. Because why this plasmid used as a cloning vector in rDNA technology means because it has three essential components. It has three essential components. This plasmid has three essential components. Hence, this plasmid used as a cloning vector in rDNA technology. Now we'll discuss what are these three essential components the plasmid it has. The first and most important is it has origin of replication, O site, or origin of replication site it has. So this site provide this specific sequence from where replication start. So here it is the essential components of the plasmids, and this specific sequence of this specific from where the replication will be start. So the replication start from this place only, these sites only, and not only that, and this ORI site or origin of replication also responsible for controlling the copy number, very important. This site only which control the copy number of linked DNA or desired DNA, of linked DNA or desired DNA. So here copy number means the number of plasmids present per cell is called copy number. Here copy number means the number of plasmids present per cell is called copy number. So this number of copy number with under the controlling of this ORI site. And one more important component the plasmid it has, so that is selectable marker, SM, selectable marker, selectable marker. Here selectable markers helps in identifying and eliminating non-host, very important, non-host, and also selectively permitting also select to growth of transparent host, Here non-transparent and transparent means simply if the host which gain recombinant DNA, such host is called transparent host. If the host do not have any rDNA in which the host are called non-transparent host. For example, here if we create rDNA, recombinant DNA, and if we insert this rDNA into host organism like bacteria, if this rDNA will this rDNA will enter into a host organism 1, then this cell is called transparent host. If this second host cell, it do not allow any rDNA, then this cells is called non-host. Means non-transparent host cell, they do not have any rDNA within it, but transparent host has rDNA with it. So these selectable marker which identify and which eliminate what this non-transparent host cell, and also we select the growth of transparent host cell only. So that is the important role of this selectable marker, very important. And for example here, and there are some examples which is given as a selectable marker. Here I am writing. Yeah. So example for selectable marker in E. coli, Escherichia coli bacteria, the genes encoding resistance, encoding resistance to antibiotics. For example, such antibiotics are like ampicillin, antibiotic resistance, and tetracycline antibiotic resistant, and chloramphenicol antibiotic resistance, and kanamycin antibiotic resistance. So all these antibiotics are considered as a useful selectable marker. All these antibiotics which consider as a useful selectable marker. Useful selectable markers. So this is all about the Escherichia coli and their selectable marker. Now we will move on. Another important or essential component of plasmids. So that is cloning sites. Cloning sites. So these cloning sites also called recognition sites. Recognition sites. So it is the region where the restriction enzyme here. This cloning site or recognition sites is the region where the restriction enzymes, that is restriction endonuclease enzymes which cut the DNA and insert foreign DNA. For example, here we have plasmid like this, circular double-stranded extra chromosomal self-replicating DNA is called plasmid. So this is the structure of plasmid. So here in this structure of plasmid, we have three essential components. Comments like this. So here we have one site. Here this site is called ORI site, origin of replication site, and here one more we have this side, and this side, like antibiotic resistant genes, like they are called selectable marker. Here we have selectable markers. For example, ampicillin and tetracycline selectable markers we have. And also one more important structure we have. So here, for example, various places we have cloning sites. Cloning sites are recognition sites, and these sites which helps in this site only the restriction endonuclease enzyme which cut this DNA strands, and instead of here, if cut means there is a space, and in this space the foreign DNA or desired DNA will be inserted, and then it forms a new combination of two DNA, that is rDNA. Okay. For example, if we remove this cloning site by using restriction endonuclease, then if we added this foreign DNA this is foreign DNA or desired DNA into this cloning site, then it become form a new DNA. That new DNA is only nothing but rDNA. So this cloning site is also very important for this plasmids. So hence, so by having all these three important essential components to this plasmid, hence it is used as a cloning vectors or carriers in rDNA technology for biotechnology purpose. Okay, so this is all about the why we use plasmid as a cloning vector, means because having all these important characters. Now we move on another important concept. So there are normally E. coli cells, they do not carry any resistance genes against them. Only we can only insert. Here note is normal E. coli cell, Escherichia coli cells, they do not carry any antibiotic resistance gene. Only we should only add this. We should only add this antibiotic resistant gene. So hence there are scientists, they created some artificially restructured plasmids. So in that we will discuss one important artificially restructured plasmids here in as per NCERT given. So that is PBR 322, and one more important, that is PUC 19 and PUC 101. All these three are artificially restructured plasmid. Restructured plasmids. So restructured plasmids means in which plasmids only we have to create some, in which plasmids only we have to add some antibiotic resistance gene to this. And here very important, here PBR is the most commonly used artificially restructured plasmid for rDNA technology. And here P means plasmids. Here P means plasmids. Here in PBR, B means Bolivar is a scientist. There are two scientists, they restructure these plasmids. Hence, or their name only, this name will be okay, Bolivar. And here R. R means, sorry, here R means Rodriguez. Rodriguez. Okay. So here PBR means P means plasmid, B means Bolivar, R means Rodriguez. This Bolivar and Rodriguez, these two scientists, they discovered these plasmids. And here 322 means the strain or the serial number of this plasmid. Serial number of this plasmid. Here coming to second and third, P means plasmid, UC. Here UC means University of California, and in which there are two series was discovered. They are 99, 101. So this is all about the plasmids. They restructured by different scientists and to added various recognition sites as well as selectable marker. Now we move on very important types of plasmids. According to function, according to functions, we have different types of plasmids. Different types of plasmids. We will discuss each one in detail. Yeah. So first one, F-Plasmid. F-Plasmid also called fertility factor. Fertility factor. Yeah. So this plasmid is responsible for bacterial conjugation process. Bacterial conjugation process between F plus bacteria and F minus bacteria, means donor bacteria and recipient bacteria via sex pili. Sex pili. And here in this F-plasmid, if the host organism has plasmid, then it is called F plus, or also called donor plasmids, or it's the male bacteria. Male bacteria. If the host organism they do not have any plasmids, and such host organisms are called F negative or recipient bacteria, or it is a female bacteria. Female bacteria. So during the conjugation process from male bacteria, one strand of plasmid, actually plasmid is a double stranded. So during the conjugation process between F plus and F minus bacteria, there is a tube will be formed. So this tube is called conjugation tube. Conjugation tube. And through via this conjugation tube only, the one of the strand of plasmid which travel and which move here like this, which move into F minus bacteria or female bacteria. So here the F minus bacteria later after conjugation process it become F plus bacteria. So like that they exchange their genetic material between F plus and F minus bacteria by this conjugation process. For this conjugation process there is a plasmid is required. The plasmid is only nothing but F plasmids or fertility factors. Yeah. So next we move on second important plasmid. Yeah. So that is R-Plasmids. R-Plasmids. Here R means resistance plasmids or resistant factor. Resistance factors. So this, sorry, one important point I forgot. Here this F plasmids contain gene for fertility. This contain gene. It contain gene for fertility. Hence this plasmid is called fertility factors. Yeah. Here R plasmid are resistance factor, and these contain genes contain genes that give bacteria resistance, very important, that give bacteria resistance against antibiotic. For example, there are two important antibiotics resistance. There ampicillin antibiotic resistance gene they contain, and tetracycline antibiotic resistance genes. This most common use of antibiotic resistance genes. And also we can use chloramphenicol and kanamycin antibiotics. So all this about R plasmids. Now coming to third important plasmids, that is cold plasmids. Yeah. Third one, COL plasmid. Cold plasmids. Here cold plasmid means cholecidogenes plasmid. Cholecidogenes plasmids. So these plasmids contain gene here. Contain genes that code for bacteriocin. Bacteriocin or colicin toxin. Bacteriocin or colicin toxin. Here, these colicidogenes plasmids which contain genes, and these genes code for bacteriocin or colicin toxins. Bacteriocin are also called colicin toxins, and these toxins which kill, very important, these toxins which kill other closely related bacteria, which kill the other closely related bacteria to reduce competition. If any bacteria which having competition with this bacteria, then these genes which contain whole plasmids which secreted, which release a protein, that protein has a toxic nature, and the toxin is bacteriocin or colicins, and these colicin or bacteriocin toxins which kill the other closely related bacteria to reduce the further competition with the competition. So that is the importance of this cold plasmids. Now coming to fourth one, so that is Ti plasmid. Ti plasmid. So this Ti plasmid also called tumor inducing plasmid. Tumor induced factor. Tumor induced factor or plasmids. Yes. And these contain tDNA. tDNA. This tDNA code for tumor inducing in dicotyledon plants. Very important. Dicotyledon plants. And these Ti plasmids, which is isolated from a bacteria, is called Agrobacterium tumefaciens. Agrobacterium tumefaciens. Here, these plasmids which induce tumor in dicotyledon plant. And in RDNA technology, we use this Ti plasmid for gene transfer, means cloning, DNA cloning. Because of this, if we disarm these Ti plasmids in Agrobacterium tumefaciens, then we can use this as a natural genetic carrier or cloner. So here Ti plasmid, generally it is a tumor inducing plasmids in plants. If we disarm means we need to remove the pathogenicity of these Ti plasmids, then we can use them as a cloning vectors for RDNA technology. So this is all about the Ti DNA. And one more important plasmids here we have, that is degradative plasmids. Here, fifth one, degradative plasmids. Degradative plasmids. And these plasmids contain genes here. Yeah. Degradative plasmids contain genes for degradation of some compounds. Some compounds. Like toluene and salicylic acid. Salicylic acids. And here, generally, these degradable plasmids are found in Pseudomonas putida. Pseudomonas putida. So this Pseudomonas pituita also called superbug. Superbug. Why these Pseudomonas pituita are called superbugs means it has a nature, means it contain a gene, it contain a plasmid that is degraded to plasmid, and that plasmid has a nature to degradation of heavy metals or different compounds, and these hence these Pseudomonas pituita are used in bioremediation process. Bioremediation process. Here bioremediation means to remove the pollutants. So solution for pollutant is called bioremediation. So for bioremediation purpose we use this Pseudomonas pituita because of this bacteria has a degraded plasmid, and that plasmid contain gene for degradation of these compounds, and hence the Pseudomonas pituita generally used for degradation of to remove this, helps remove of various compounds like oil split, oil splits, and also which remove plant wastages, and also which remove toxic wastages. So to remove all this we use Pseudomonas pituita, hence the Pseudomonas pituita is known as super bug. So all these nature because of that plasmid has a degraded to plasmids. Right. And last and most important, so that is weir plasmids. Yeah. Here sixth one, weir plasmid. So this plasmid contain genes, for disease causing, for disease causing. Hence this weir plasmid, hence this weir plasmid is known as virulence plasmids, virulence plasmid, or pathogenic plasmid, virulence or pathogenic plasmids, and disease caused plasmids, disease caused plasmid. So for example here, for weir plasmid example, so here Ti plasmid and one more plasmid, that is Ri plasmid, Ri plasmid, Ti and Ri plasmids both are example for weir plasmids. Here this Ti plasmids which present, which isolated or which is present in Agrobacterium tumefaciens, Agrobacterium tumefaciens, tumefaciens. And here Ri plasmids which is present in Agrobacterium rhizogenesis species, Agrobacterium rhizogenesis species. And this Agrobacterium tumefaciens which cause crown gall disease in dicotyledon plants, crown gall, crown gall disease in dicot plants, and this Ri plasmid in Agrobacterium rhizogenesis which leads to cause a disease called root hair root hair root hairy disease. It causes root hairy disease. So likewise this bacterium like Agrobacterium tumefaciens and Agrobacterium rhizogenesis species both having weir plasmids that contain genes for disease causing. This Ti plasmid cause crown gall disease in dicotyledon plant. Ri plasmid cause root hairy disease. If we disarm these Ti plasmid or Ri plasmids of their pathogenicity, and then we can use this Ti plasmid or Ri plasmid in RDNA technology. So this is all about the different types of natural plasmids based on their functions. And also we discuss one important artificially restructured plasmids like PBR 322, PUC 19 and PUC 101. So this is all about the entire concept of plasmids and its structures. Now we have three important MCQs from this topic. Yeah. Yeah, it is the first MCQs from this topic. So read this statement. Which of the following statements correctly defines a plasmid? So here asking about definition of plasmids. Okay, read these four options. And one more MCQs. Yeah, second MCQs here. Yeah, a specific type of plasmids contain genes. That genes code for toxin. Here toxin is called colicins. Just now we have uh, discussed about colicin toxins, or also called bacteriocin toxins. Colicin toxins. And which functions to kill other susceptible bacterial cells. Susceptible, sorry, suspectable bacterial cells. Identify this type of plasmids. Yeah. And last MCQs here. So in the context of bacterial plasmids, what does the term copy number refers? Copy number. Here read for this option and then answer your answer and drop your answer in comment box. So thank you. Have a nice day.